Okay.
Yeah. Good afternoon, all. I welcome you all for session 3 for the day. We have with us Dr. Archana Kanawade ma'am. Ma'am has a PhD in Electronics and Telecommunication from Savitribai Phule Pune University, her Masters in Electronics and Communication from Shivaji University. Ma'am has around 17 plus years of experience. Her expertise include MATLAB Signal Processing Toolbox, R Programming Language, C Language, Arduino Programming, Weka Toolbox, Biomedical Signal Processing, and many more. Ma'am has a thesis titled Design and Development of Non-Invasive EMG Analysis Technique for Chronic Obstruction Pulmonary Disease (COPD) in the area of biomedical signal processing. She has a book published on electromyographical signal analysis and is accepted for publication by elsewhere. She has published around 35 papers in national and international journals and conferences. Ma'am has guided a number of master's and bachelor's projects as well. Ma'am has various proposals submitted under CSIR Research Scheme, BIRAC Biotechnology Industry Research Assistant Council, BCUD, BST Biomedical Device and Technology Development Program, AICTE, RPS Research Promotion Scheme, and Big BIRAC, that is Biotechnology Ignition Grant. Ma'am, on behalf of JSPM's Rajarshi Shahu College of Engineering and Department of Electronics and Telecommunication, I, Shraddha Bindhargive, welcome you, ma'am.
Thank you, Shraddha. I really thank you, Bisikar Madam, Paithani Madam, HOD Zadav Sir, Paithani Sir, for sharing my knowledge on the EMG processing on this platform. And thankful for your wonderful introduction, Shraddha. So I'm sharing my screen and directly starting with the presentation. Welcome all. Yes. Yes, welcome all of you for this electromyography, that is EMG, signal processing technique. Myself, Archana Kanawade, these my phone number and my email. So here in this topic, basically in electromyography, I'm going to start with the acquisition of electromyographical signals, and after that we are going to discuss the signal processing techniques, that is time domain analysis, frequency domain analysis, and time frequency domain analysis of the EMG signals. Let's start. First is, what is electromyography? So electromyography is a electrical manifestation of the neuromuscular activity of the muscle signals. Whenever we are capturing the signals from the muscles, it is an electromyographical signal. If it is a heart signal, then ECG. If it is a brain signal, then it is EEG. But whenever it is muscle signal, then it is EMG, that is electromyography. So this is your motor neuron. Here we are having this motor neuron. Brain sends the signals through this motor neuron towards this axon. Axon passes this signal towards the muscle. So here we are having the neuromuscular junction, which are the end plates. Signals from this brain, from these neurons through the axon will be passed to these end plates, which are connected with the muscle. In muscles, what we are having, it is a individual muscle fiber. Inside this muscle fiber, there will be muscle cells. So what happens whenever there is an excitation, this innervated muscle fiber membranes give Na plus ions to flow that results in the depolarization. So whenever there is a depolarization because of the flow of Na plus ion, there are active ion pumps which restore the ion in equilibrium by doing the repolarization. So this depolarization process and repolarization process spreads the action potential throughout this muscle fiber from both the directions, that generates the electromagnetic. And whenever this electromagnetic field we detect with the help of electrodes, we call it as a electromyographical signals. These electromyographical signals, it basically controls by the central nervous system. How it controls? It controls it by the number of recruited motor neurons as well as by using the firing frequencies. This EMG is dependent on multiple factors, means anatomy of your muscle, muscle fiber diameter, length of your muscle fiber, physiology of the muscle, age of your muscle, peripheral nervous system which is associated with the muscle, as well as technique of your detection. EMG varies with the technique of detection as well. EMG varies with the temperature as well. So if you're capturing the EMG signal at 24 degrees Celsius, it will give you some different amplitudes and the frequency. If it is captured at the 30 degrees Celsius, it will give you different. So whenever you are capturing or acquiring the EMG, temperature, your body temperature should be maintained constant. So these 6 points are very, very important while acquisition of the EMG signal. Now next is the electromyographical signal generation process. So we have studied this. These are the motor implants. These are your nerves, motor neurons. These are the implants, and this is your individual muscle fiber. So inside a single muscle, there will be n number of muscle fibers. This is your first, second, up to this, suppose, and n number of muscle fibers. So here, your potential, that is repolarization and depolarization process, will be like this. So it will be captured like this. At this point, it will be like this. At third muscle fiber, it will be like this. And at this point, it will be like this. But whenever, whenever we are recording these electrical changes in the muscle, electromagnetic changes in the muscle, through the recording sites. The recording site means what? These are your electrodes, 2 electrodes. Whenever we are capturing these signals, so it is a total superposition of the action potential contributed by individual muscle fiber. So this signal that we are acquiring is a superposition of action potential contributed by a single muscle fiber. So this also what it depends on? It depends on the diameter of muscle fiber, distance between the active muscle fiber and the recording site. Means this is your recording site and your muscle is, suppose this is a surface EMG. So at that time this recording site and your actual muscle position will be different. If it is a invasive, means needle electrode also, at that time also this recording site and actual muscle fiber position will be different. This distance between this recording site and the actual muscle also contribute towards the change in the, is dependent on the this distance between the active muscle fiber and the recording site, as well as the filtering properties of the electrode. What kind of electrode you are using over here? It might be needle electrode, EGCL electrode, EG electrode. So depending on which type of electrode you're using, your EMG changes, EMG amplitude varies. So this is a basic acquisition. Any biomedical signal acquisition, it goes through like this. First, this is a muscle. So this is muscle signal. There will be one reference. Reference in whenever acquiring the electromyographical signal, placement of this reference electrode is very, very important. So where you can place this reference electrode is a position that you have to identify where we are having the less muscle or the zero. We are having actually a bone over there inside your skin. At this position, you can place this reference electrode. There should be no any zero contact with these muscles. So we can have your this forehead. You can place this place. You can place this reference electrode. On legs also we are having different places where there is directly bone is coming inside the skin, and there is very less interference by the muscle. At that position, you can place this reference. So here, whatever the signal acquired from this electrode is amplified differentially. Here you can use the instrumentation amplifier, which provides the gain as well as it provides the differential gain. And you can use the high pass filter to remove the high frequency, unnecessarily high frequency which are captured, as well as you can use the low pass filter to remove the low frequency band. And one important thing is you can use a notch filter. So these analog signals, after that it will get converted into digital. So ADC, for your normal operations, your ADC requirements are different. So here you should have the maximum bit of 12 bit or greater than 12 bit, 16 bit ADC is required. It will be given to the PC for the signal conditioning. We hear differential amplifier, high pass filter, notch filter, low pass filter, and analog to digital converter. Now, whenever designing this high pass filter and these low pass filter, points to be remembered that for every muscle that you are selecting for your study, its frequency of operation will be different. So if you are choosing, suppose your skeletal muscles are there, its high pass filter, its cutoff frequency will be different. Its low pass filter cutoff frequency will be different. So depending on the muscle of your study, you have to choose these cutoff frequencies of the high pass filter and low pass filter. In my study, I have used 240 hertz of the frequency was there, cutoff frequency, and low pass filter. Uh, I haven't done the low pass filtering as my most of the data was accumulated in the low frequency. So depending on your study, depending on your muscle, you have to choose these cutoff frequencies of this high pass filter and low pass filter. Now, whenever making this acquisition circuitry, again, some more points that you have to remember is the skin preparation is very, very important. This electrode contacts with this here with the skin should be very, very stable. Why? You should get at minimum in order to get the minimum skin impedance. Minimum impedance is very high. So after applying the conductive gels or using the stable electrodes, you can minimize this skin impedance. Electrodes also should be chosen which should have the high signal to noise ratio. In which posture you are acquiring your signal is also has to be decided, as well as a reference electrode position. Like I have said, the reference electrode position, there should be a immediate contact with the bone and zero muscle uh, interference should be there. Temperature should be kept constant. This is very necessary criteria because with temperature your amplitude of the EMG changes. And stabilization of the chemical reaction, this is also very important point. After application of the conductive gels, means while doing the skin preparation, you have to stop for certain minutes, for 1 minute or 2 minutes. What happens because of this conductive gels, impedance of your skin lowers, and whenever we are having the lower skin impedance, you should acquire your signals in order to get the maximum EMG signal acquisition. Okay, so these are some recording techniques that we are going to discuss. As discussed, electrode detection surface should have the low impedance, should have the large detection volume. Large detection volume, if it is greater, then you will get the larger, more EMG signals will get acquired. You will get the minimum distortion as well as you will get the highest signal to noise ratio should be there, whatever the electrodes you are. There are different recording techniques. First is the surface EMG, which is non-invasive. Means on the surface of muscle, you are placing the electrode. We are not pricking inside the body. Surface EMG is very beneficial. If you want to capture the signals from the some peripheral outside muscles, and it is very easy to get the consent from the patients. That's why surface EMG is important. Otherwise, patient generally doesn't argue for this intramuscular EMG, that is for the invasive technology. And in surface EMG can be acquired by using the two types of electrodes. One is the dry surface EMG electrode, and second is the gelled surface EMG. Dry surface EMG are nice electrodes. These are passive electrodes. They are heavy, but whenever you will capture the signal in the sitting portion, it possesses electrode fixation problem due to their heavy weight. They are heavy, so you have to add the adhesive tapes over there for this dry surface EMG electrodes. And these gelled surface EMG electrode are lightweight and passive. Next is the intramuscular EMG, that is IEMG. That is, but this technology is invasive. So where we use this invasive technology, we capture the good signals through this invasive technology, intramuscular EMG compared to the surface EMG, because intramuscular EMG is in directly contact with the muscle. So this distance between your muscle and the capturing surface is lesser in the intramuscular EMG. Because electrode needs to be inserted inside the deep skin, but it requires the prick has to be taken. Patient generally doesn't give, or the subject generally doesn't give the consent for this intramuscular EMG. But whenever we are studying the muscle, which is light inside a body, deep inside, located deep inside the body, we cannot use surface EMG. Like for diaphragm signals, like your scalenus muscle, which are the muscles which are very small and inside, located deep inside the body, we cannot use the surface EMG signal. Now the third type of recording technique is EMG triggered stimulation. Here, patient has to, this generally happens, EMG triggered stimulation. We studied this for the myopathic and neuropathic conditions where patient initiates the muscle contraction. And whenever the specific EMG threshold is reached, at that time, the muscle is stimulated externally to complete the contraction. These generally EMG triggered stimulations and do it for the myopathic and neuropathic conditions. In some conditions for the healthy muscles also, this EMG triggered stimulation is performed. Now, coming towards the noise which are we can see inside this EMG. So first is the inherent noise in the electronic equipment. So if your equipment design is very proper, it's whatever the components you are using or the filters that you are designing is very proper, then this noise can be not can be eliminated but can be minimized. Inherent noise, ambient noise of 50 hertz power line with the notch filter, good design of notch filter can be removed. ECG artifact. So whenever most of the times happens, your muscles signals, we have our pulses that can also be detected nearby to your muscles. We are having, so these ECG signals also get captured. I face this problem a lot. ECG signal also get captured with the muscle signal. Here, what you have to do, placement of your electrodes, whatever the muscle that you have to study, its placement of the electrode plays an important criteria over here. So off axis placement of the electrode can remove this ECG artifact. Next is the motion artifact. So there is we are having some motions. Patient cannot be still stand like this for your whole duration of the study. So this motion artifact also comes in the picture. That is that can be removed by the firm placement of the electrode and by using the low pass filtering. Now, next noise is the DC offset. It occurs due to the difference between the electrical impedance in the skin and the electrode. Which is said earlier that the proper impedance matching, having a good input impedance and a application of conductive gel. Application of conductive gel, and after application of the gel, you have to wait for 1 minute so that this impedance will get lowered, and you can get reduced of this DC offset noise. Next is the muscle crosstalk. Human body is not like that. Whenever there is a one muscle, there will not be any association of any another muscle. So there might be capturing, while this capturing of the signal, there might be a muscle crosstalk signal, that is a signal from another muscle. It is not always like that we are studying this muscle and we will capture the signal from only that muscle. So proper placement of the electrode at the center of muscle belly can reduce this muscle crosstalk signals. The recommended settings for your EMG acquisition is CMRR should be greater than 100 dB. Input impedance should be higher than 100 mega ohms, and 16-bit analog to digital converter or greater than that can be used for capturing of the, for acquisition of the EMG signals. This is EMG acquisition or the raw EMG signals. So this was my placements of the electrodes, my study. This is a sternomastoid muscle. So here these electrodes are placed at the center of the muscle, positive, negative, differentially amplified. Reference electrode is placed on the forehead. We are having the less interference from the muscle. And this is your raw EMG signal, which is in time domain, 480 samples per seconds. Here you can see this muscle is active in this region, and there is a deactivation of the muscle in this region. This is your total muscle activity. Here the muscle is active and muscle deactivation. Okay. So here the instrument used was RMS Salas, these 16-bit input impedance. Slab of alcohol is used for the skin preparation, and temperature was kept constant at 24 degrees Celsius, and the signal is acquired in a sitting posture. And this protocol that we have to use is a respiration. This is the actual data collection. This is a machine, RMS Salas machine. We are having these electrodes. See, you can see these electrodes. This is a 2-channel machine. This also works as the NCV machine. And these are the placement of electrodes. See, these adhesive tapes are used because this is a reference position of the electrodes. Now see the surface EMG signals. Sometimes you can see this clear activity of the muscle over here, but sometimes here the muscle is not active, you are getting this kind of signal. Here the muscle is active, still you are getting this kind of signal. And here the muscle is not active, still we are getting this kind of signal. Here the EMG analysis in such cases is very, very simple. But while we are getting such kind of signals, at that time analysis of the EMG either in time frequency or time frequency domain is little difficult. So now coming towards the EMG analysis, second part. So first, before, as I told you earlier, before doing any kind of muscle analysis, first you should know the muscle conditions. The muscle that you are analyzing, it is whether it is a healthy muscle, whether it has a myopathic conditions, or whether your muscle is having some neuropathic conditions. So if you are analyzing the muscle, this muscle which is having the neuropathic condition, you may get wrong results. If your muscle is having the myopathic condition, you may get wrong results. So before analyzing any muscle, you should confirm that it is a healthy muscle, or if you are specifically studying the healthy myopathic neuropathic conditions, then you should do it. You should do the comparison. But the muscle should be a healthy muscle for your general analysis. But what is the neuropathic condition and what is myopathic condition? Neuropathic condition means there is a problems in the neurons. Muscle is absolutely well. There is no problem in the muscle. But the neurons which are sending the signals, brain is sending the signals through the neuron axon towards the end plate, towards your muscle fiber. So there is a problem in that total structure. And because of that, brain has to send the excess signals, excess amplitude signals, excess frequency signals, that is called as a neuropathic condition. You can see, observe the amplitudes are more as well as the frequency is also greater. In a myopathic condition, what happens? There is axons, your neural conditions are very nice, but your muscle fibers, they are damaged. The muscle fiber inside your muscle, the fibers are, there is a damage in some muscle fibers. So that condition, you will get this kind of signal, which is called as myopathic arrangement. So for normal, you should analysis of your signal for general purpose, it should be healthy signal. But you should find that whether your muscle is not in the neuropathic condition or in myopathic condition. This is again a healthy myopathy and neuropathic condition. Myopathy condition, neuropathy and healthy after full wave rectification. If you see the frequency domain analysis, here you can have the myopathic conditions. Very larger frequencies are used, amplitude of greater than the 100 in the frequency domain, while it is in healthy and in myopathy, they are the same. After identifying this myopathic and neuropathic conditions and the healthy conditions, normalization of the muscle signal is also very important. The normalization, if you see, this is mean amplitude of the EMG signal, and this is a person's height in centimeter. So as height increases, mean amplitude also increases. So this normalization, here the signal is normalized now. There is no dependence of the height. So this is a normalization. So if you observe your signal, person's height increases, EMG amplitude decreases. If a person's, if it is a male subject or the female subject, at that time also the amplitude of your muscle, it differs. As well as you can age of the person also matters. Greater aged person, its amplitude differs. Lesser aged person, its amplitude differs. As well as the working condition of the particular person. Working condition of the particular person. If that person is using that muscle multiple times, then also its amplitude differs and its frequency differs. Okay. So all these things should be normalized, or you should do something for that difference in the amplitudes and frequency. Now, so first what you have to do, either you have to identify the myopathy, neuropathy and healthy conditions, then you have to normalize your signal. After normalizing, then you can go towards the analysis. First we'll start with the time domain analysis. So this is your raw EMG signal. See, this is actual signal. This is the raw EMG. This is full wave rectified EMG signal. So here, this full wave rectification, linear envelope, that is the envelope detection of your muscle is signal is also very important, as well as the integration is also very important feature. So what this linear envelope exactly gave you, so this full vector rectified EMG signal you have to pass through the low pass filter will give you the linear envelope. So linear envelope will give you the profile of the EMG signal, which gives the duration of the muscle activity. So this is the total duration of the muscle activity you can identify. This is also a duration of the muscle activity. But if you want to analyze the profile of the EMG signal, then at that time you can go for the integrated EMG. So linear envelope and integrated EMG is two basic time domain features that you can analyze. The next features that you can extract from this time domain EMG signals is a mean. Mean of the signal will give you total activity of the signal. So this is your total mean of the signal. After that, there is another feature which is called as average rectified value. Here what happens, average rectified value gives the directional activity is given the equal weightages in the ARV. Next feature is the root mean square value. Root mean square value is a non-linear detector. It gives the direct relationship with the power spectrum, and mean give the inaccurate information about the fatigue analysis. If you want to analyze the power of your signal, you can go for the root mean square value. Most of the researchers, they use the root mean square value. But whenever you want to analyze the muscle fatigue conditions, then this gives the inaccurate information. Variance is also one important factor which measures the variability in the signal. So according to my point, this variance is also one important feature. I got a lot of information from this variance feature also. Next feature is a time domain feature is a zero crossings. It is very simple feature, zero crossing. Number of times your signal is crossing zero. It is very simple, but whenever you will not find any solution or any information from this RMS value, this mean value, from this your average rectified value, at that time this zero crossing gives you very important information. So this zero crossing is also very important feature. Wherever your signal is crossing zero, from positive side to the negative side will give you the zero crossing. In this figure, your total zero crossings are 64. So with this zero crossings, again another feature comes, that is a number of zeros. In your EMG signal, if you observe multiple times, you will get the signal, you will get the 0 0 0 0. If DC offset is not there, you will get 0 0 0 0. So how many number of times you are getting the zeros in your signal is also one important feature that also gives multiple times your very good information. Next feature is conduction velocity. Very few researchers has used this feature, that is a conduction velocity, but this is also a very important feature. So here what happens in the conduction velocity, at what velocity your muscle conducts these electrical signals or through neurons signals are coming. This is a conduction velocity. So here first you have to analyze the number of peaks. This is your number of peaks which are there in the signal. See this diagram. These are the number of peaks. And for these number of peaks, after that what we are doing? We are finding what is the conduction velocity for this number of peaks. That is from this point to this point, your signal is going. From this peak to this peak, your signal is going. But at which speed it is going? But at which speed it is going can be calculated through this conduction velocity. So this velocity of the conduction is calculated over here as one feature. If you go for the NCV test, that is, it is an electromyographical type of test only, that is an NCV, that is a nerve conduction velocity. So at that time, on the body surface, on the muscle, they will have the 2 points where the signal will get captured to the electrodes, and you have to capture whenever the pulse is passed and whenever it is reaching at the first point, and after that, after how much time it is reaching at the second point on the muscle. Because this is your muscle, here at this point your signal will get transmitted, and after what amount of time your signal will get transmitted over here is also called as conduction velocity. That is called as nerve conduction velocity. This conduction velocity and that conduction velocity is different. This is conduction velocity of the surface EMG. These are the total maximum time domain features that I have accumulated over here. Maximum amplitude, minimum amplitude, peak to peak amplitude, mean amplitude of your signal, average rectified value of your signal, variance in the amplitude is also one important, number of peaks over here. See this formula. So here amplitude change 0.1% of the maximum x of I. So why this 0.1%, 0.1 is multiplied over here to avoid the unnecessary noise which is introduced in your signals. This 0.1 is introduced. Root mean square, skewness, entropy, kurtosis, zero crossings, number of zeros, as I have shown in this diagram, conduction velocity. This conduction velocity, we can have the average conduction velocity as a feature, as well as you can have the maximum conduction velocity also as a feature. So while deciding the features, you should consider the very important conditions or the things that you want to acquire from the muscles. Okay, this whatever the information that you want to acquire from the muscle, depending on that, you have to choose your time domain. Okay, so this is just a time domain feature extraction from the raw EMG signal. But while you are discussing about the time domain analysis, one important thing comes in the picture, that is onset detection. Onset detection. So what do you mean by onset exactly? Onset is the on-off timings of the muscle. So here you can observe in this raw EMG, your muscle is active over here. Muscle is active over here and muscle is not activated over here. In this region, there is no muscle activation. In this region, no muscle activation. Muscle is active only up to this point. So whenever you want to see only, you want to check only activation signals, once muscle activity signals, whenever you want to study, at that time, this onset detection algorithm comes in picture. What is it? Onset detection algorithm is on-off timing detection. This is on timing, this is off time. On-off timing detection of the muscle is an onset detection algorithm. So onset is what? Onset is initiation of rapid motor response. That is, initiation of this activity is called onset, and end of motor activity, motor unit activity, is called as offset. Okay. So if we'll detect this onset and this offset timing, if you detect the onset and offset timing, it makes the processing of the EMG easy and accurate. Why? Because your time duration, it reduces. Your number of samples reduces. Your area of analysis, it also reduces. And as well as features that you'll capture, features that you are getting after doing the onset detection algorithm, running the onset detection algorithm, you will get the improvement in the features. Okay, so features acquired during the active duration of the muscle are very distinct, and length of the EMG reduces, it reduces the processing time and number of computations. Let's see some onset detection algorithms which are there in the literature. First basic algorithms is first is the single threshold algorithm, and second is your double threshold algorithm. So in single threshold algorithm is what? By checking the your noise in the muscle, this threshold is detected. This threshold is detected by considering the noise in the signal. So suppose this is your threshold. So whenever it is crossing the threshold, whenever your signal is crossing the threshold, it will be detected as onset. And whenever the last point it is crossing the threshold, it is detected as a offset. So here, this is single threshold algorithm. You can see the threshold over here is a red line. So this is a threshold. So this is here, it is detected as onset. This is the second time your threshold is detected as offset. So here in single threshold algorithm, this total area is totally detected as a active region of your muscle. But here what happens? This is actually a noise. This is also a actual noise. Active muscle signal is only from this duration to this duration. So this is a limitation of single threshold method. Single threshold method is very simple, but there are chances of errors detection of the onset and offset instant. You can go for the double threshold method. In double threshold method, wherever it is crossing the threshold, there will be a pulse generated. And whenever that pulse is having, that this is your second threshold, is having a width greater than your cutoff timing, greater than the cutoff timing, will be detected as onset. You can see this will be discarded, this will be discarded because it is not crossing this TC. This pulse is also discarded because it is not crossing the TC. So here at this point, this is detected as an onset and this point is detected as a offset in double threshold method. So this is your onset and this is your offset. This is your total active region. This is your total active region of the muscle signal. So this is your some onset detection algorithm. So whenever you want to go for the time domain analysis, specifically for the active duration of the muscle signal, whenever the muscle activity analysis you want to do, not deactivity. Yeah? Whenever you want to go for the muscle activity detection at that time, I recommend you should go for the onset detection algorithm. You can use single threshold, double threshold, or I have designed my own onset detection algorithm that you can use, or in literature you can find multiple onset detection algorithms are there that you can use. The onset detection algorithm that I have developed is called as slope based onset detection algorithm. It is a slope based onset detection algorithm where the slope of the individual signal, first the signal is segmented with a window size of 20. This window size, again window size of 20 is decided by doing the analysis for the window size by analyzing its RMS values, signal RMS values, noise. This window size is decided, and for this window size, the signal is average instantaneous amplitudes calculated, and for whenever we are getting the highest slope is detected as the onset and the lowest slope is detected as the offset. I'm getting very better result compared to the single threshold and double threshold method, and number of competitions has reduced a lot in this slope based onset detection algorithm. So what I recommend, that whenever you want to analyze the active duration of the muscle signal or activity of the muscle signal, you should always go for the onset detection algorithm. Either you can develop your own, or you can use the algorithms which are recommended by the literature. So this is a slope-based onset detection algorithm. So here this average instantaneous amplitude, and this is a threshold. Now, what this slope-based onset detection algorithm will give you the onset and offset duration. And if you'll calculate the time domain features once again, after application of this onset detection algorithm, definitely there will be a lot of improvement in the feature values. You can observe this. I have experience, you have to experience. Next is the frequency domain analysis. Uh, Again, time domain signals doesn't give total information about the EMG analysis. You have to go for the frequency domain analysis. Excuse me. Time domain analysis only will analyze the amplitudes, but at what frequencies neurons are sending signals to your muscle, and what is the effect of this frequency on the fatigue, especially the fatigue analysis. If you want to analyze the muscle fatigue, that cannot be done by using the time domain analysis, because time the amplitude of the signal, EMG signal, decreases after getting the fatigue analysis, after muscle fatigue. In frequency domain analysis, you can have the mean frequencies and median frequencies. That mean frequencies and median frequency gives the good analysis of the fatigue of your muscle. So frequency domain analysis we can do by using the fast Fourier transform, or the oldest method, by using the F50. Here you can analyze the mean frequency, median frequency, as well as power spectral density analysis. Most of the researchers, they do the power spectral density analysis as well. What happens? Area under the power spectral curves reveals the total power contributed by your muscle. So as power increases, contraction force increases, and the power spectrum shows a smaller shift towards the upper side of the spectrum. So this is the power spectral density. Spectrum at a specific window length. Like in onset detection algorithm, we have divided our signal with a window length of 20 samples, window length of 15 samples. Here also, you can divide your spectrum, your EMG, into a specific length of segments, and that segments you can analyze individually. That also can be done. In the time domain, we have analyzed the onset, we have calculated the onset, as well as we have calculated the offset. So at onset, certainly at a certain point, your amplitudes are increasing at a greater rate. And at offset area, your amplitudes, EMG amplitudes, are decreasing at very faster rate. This frequency distribution in between the active region, at the onset area it is very different, at the offset area it is very different, and in between the signal it is very, very different. You can see the frequency distribution. So in some of the research, the frequency distribution at the onset area contributed a lot meaningful features, as well as at the offset area. So you have to do the frequency distribution. You can study the frequency distribution at the onset area also, and at the offset area also. Many of the researchers, they have also done the frequency domain analysis by using average instantaneous frequency. Like we have time domain analysis, we have used the average instantaneous amplitude for onset detection. Many of the researchers, they also use the average instantaneous frequency because it doesn't require any causal stationary assumption, and it has a less variance compared to the mean and median frequency, like compared to your fast Fourier transform. Here, but cannot be used for the sustained contraction. This method cannot be used. So this is your single-sided frequency spectrum. You can see this is whenever the muscle is activity. You see the amplitudes, your frequency spectrum, single-sided amplitude spectrum. and whenever there is a very less muscle activity, you can see this. So these are some more examples. This is some for first type of signals, for second, for third and fourth type of signal. You can see this kind of frequency spectrum, that is single-sided frequency spectrum. Now the spectral features that you can acquire is maximum amplitude, which will give you fatigue, mean amplitude, that is the average rectified value of your frequency, variance in the amplitude is also one of the important feature, root mean square, and the number of peaks which are observed in the frequency spectrum, number of amplitude peaks in the frequency spectrum will give you the conduction force. This is also one important spectral feature. Next is the power spectral density analysis. Sternomastoid activity increases. Your muscle activity increases with the severity of your signal. So, but in general, what happens in the power spectral density analysis? Your results of the PhD analysis is varies with the power spectral density estimation method. I have used the multiple power spectral density estimation methods, but every time the result is different for every method. So here, this is power spectral density calculation. This is with autocorrelation. This is the wage power spectral density estimation. You can see these two graphs. This is for the diseased person and this is for the healthy person. Here the muscle activity is more, here it is less. Again the parallelogram Birch power method, but the results are really different here. Here again these 4 lines means give you your 4 different signals. That is this is Burke method only, but the blue color is some grade 1, 2, 3, 4 and black color is some different grade. But it is very difficult to identify the different grades over here through this power spectral density. This is my observation. This next is spectrum at the window length of 15. At the window length of 15 sample, if you do the spectral analysis, you get some good pictures. But from this also what you do feature. Depending on your need, depending on your whatever the results, contributions you want from that feature, you have to choose the feature. This is feature pick. Slope can be one feature. First slope location can also be a picture. According to my for my research, the slope has contributed a lot as a feature. Next is the spectral features at the onset and offset instant. As I told to you, the onset area. At onset area, if you do the frequency analysis, it also contributes a lot. And at offset area, if you do the frequency analysis, also it contributes a lot in terms of features. So here, again, the features are calculated. Major spectrum, major spectral location is calculated. Spectrum under 50 hertz, greater than 50 hertz. This frequency also you can choose by observing your frequency spectrum. So these are some features which are selected and done the analysis. This is this is your total frequency domain analysis of your EMG signal. So here also you have to choose your method of analysis very carefully, and depending on your need, you have to choose the features. The results that whatever the way you want, what kind of results you want, you have to choose your features. Next, we are going towards the time frequency domain analysis. Excuse me. Now, only the time domain analysis and only the frequency domain analysis may not contribute for the total analysis of EMG signals. Because what happens? Because EMG is non-stationary in nature. With the muscle, signal differs. With person, signal differs. The same person at the same temperature, signal differs. If you are taking capturing a person's signals today and tomorrow, signal differs. Signal differs. So that's why it is a EMG is non-stationary in nature. There is a no repetition. Time localizations of the spectral component. It provides the important information. But the inherent variability is a major drawback of the EMG measurement. So that's why, again, what you have to go, you have to go for the time frequency domain analysis, literature survey that you have to do. So here, short time Fourier transform is the first thing. And wavelet is most of the researchers are using the wavelet analysis for the time frequency domain analysis of the EMG signal, specifically for the EMG signal. So what happens in the short time Fourier transform? The spectral components you can gain at the given time interval. In this given time interval, you can get the spectral components. At this given time interval, you can get the spectral components. And signal which are there in this window, it should be necessarily should be stationary. The stationarity analysis has to be done to find the window size. Like we have calculated it and we got the 15 samples per window. They have poor frequency resolution, while the wide window provides a poor time resolution. So selection of this window is very difficult over here in the short time Fourier transform. But if you have done with the good stationarity analysis and got the good window size, you can go for the short Fourier transformers. It works good. Now next is what? Another solution is what? Wavelet transform. So what happens in the wavelet transform? This area of the square is the same width and the height of the square changes depending on the frequency axis and the time domain axis over here. So what happens at the low frequency, height of the boxes are very shorter, which is giving the good frequency resolution, but the widths are longer, which is giving the poor time resolution, but the area is same in the wavelet transform. So here, suggested most of the researchers, they have suggested is the wavelet analysis, but you can go for the Wenger-Wille distribution. Some researchers, they have also gone for the auto-regressive moving average method. What happens in the Wenger-Wille distribution? Uses the autocorrelation function for calculating the power spectrum. But what happens? Interference handling is difficult because it appears in the signal in this WVD method. And auto-regressive moving average methods, it consider the short time intervals, but selecting the order of the filter is difficult. Some say that order of the filter should be 4, 5, some say that 20, 50. So result vary with the filter order is the auto-regressive moving average. In earlier days, most of the researchers, they have used this auto-regressive ARMA method for the analysis. Now, in wavelet also, we can do the analysis by using the discrete wavelet transform, as well as you can do the analysis with the wavelet transform. Generally, in literature, the discrete wavelet transform at the level 4 with the Davichi's 7 or Davichi's variants has found to be used by multiple of the researchers. But what happens in the discrete wavelet transform, if your sample number of samples per seconds are very, very lesser, then it degrades the performance as we are downsampling our signals. And in continuous wavelet transform, what happens? It plays an important role in the analysis, varying scale. Scale of your mother wavelet can be vary at the steps of octave, logarithmic scales, or the single frequency analysis at particular interested frequency can also be done over here in this continuous wavelet transform. So this is just a demonstration. What is the wavelet is actually, this is your signal, which is a raw EMG signal. You can see this, this is a raw EMG signal, and this orange one is your mother wavelet. So here, whenever we are having the high correlation, what happens? We are having the scaling factor. This is a scaled. This is DB7 mother wavelet at scale at certain frequency. And what happens? It overlaps. These waves overlaps from, that is translational factor is there from this point to this point, like this. It overlaps here. We have this, like this, like this, like this, like this, like this. And whenever it is finding the high correlating area, see this, whether it calculates the correlation. Whenever the high correlation area is there, you will get the maximum value of the your wavelength coefficients. And here we are having, see this point, here we are having the less correlating area. So it time stamps, it overlaps from this point to, that is a translational parameter. It is moving from this point to this point is a translational parameter. And it is widening of your mother wavelet, like widening. This is high, your lower scales and higher scales is your scaling of your mother wavelet. So this mother wavelet at particular scale has to be translated from this 0 point towards this, the maximum axis of your time duration. So whenever this mother wavelet will find the high correlating area, you will get the maximum coefficients. And whenever we are having the low correlating area, you will get the lesser coefficients. You can see this. So this is higher CWT coefficients at the maximum correlating area. And here you are getting the lower CWT coefficient at the lower correlating area. You can observe this. See here. This. So this is a continuous wavelet transform. This is a continuous wavelet transform, and this is a scaling and the translational parameters. So here this time frequency domain analysis, multiple review papers, 104, 26, 47, 42 papers has been reviewed by these peoples, and they are saying that in TWT, CWT, DWT is very good. DB2, DB6, DB8, CWT is good and DWT is good, but at level 4, CWT efficient, DWT, DB. What kind of wavelength, mother wavelet should be used for the time frequency domain analysis using the wavelet is also one important. So here what you can do, that you can do the literature survey analysis specifically for your muscle. Specifically for the muscle that you want to analyze, or the type of, means if it is a fatigue analysis, if it is a another amplitude analysis, or it is some activity analysis. So in that region, do the analysis, you can get this mother wavelet. So here in most of the their literatures, they have used the DB. So Davichis is the mother wavelet that is being used by most of the literatures for analysis of the EMG. Coeflet also and Davichis. So here or simulate by orthogonal Mayer also you can use, but DB and the coeflet is used by the most of the researchers. So here, what is, we'll start with the CWT first. That is, CWT is continuous wavelet transform. So here what we, as I told to you, we are having the two parameters. This is a mother wavelet. We are having this scale parameter, and this is your translational parameter. That is means what? Your wavelet is sliding. Your wavelet, mother wavelet is sliding on your signal is through this translational parameter. And scale parameter is you can adjust the frequency or your width of your mother wavelet. But you can scale your mother wavelet according to its frequency. It's a scaling parameter. So here, scale, how to decide the scale is also an important thing. Scale can be decided by using the dominant frequency. For this dominant frequency, you can choose, you see this. Say your center frequency is this. If your delta value, delta is what? Delta is a 1 upon the sampling rate. Whatever the sampling rate that you're using is the delta center frequency. So for your dominant frequency, you will be having different scales. So by doing the FFT analysis, you can find the dominant frequency which is there in your signal. And for that dominant frequency, you can have your mother wavelet. You can design your mother wavelet and you can do the analysis. So here, these are some dominant frequencies that I have represented, which was there in my signal. So that's why I have taken that. And for this is the scale value. And for this particular scale, you can design your mother wavelet. Or you can have, like here, You can vary the scale values at the steps of octave. If you don't have any idea about the dominant frequency, you can vary the scale according to the steps of octave, logarithmic values, or a single frequency, and you can do the analysis. So this is the dB7 at the different scale. The lesser the scale is, higher is the frequency. The S is equal to 1. This blue one is S is equal to 1, lesser the scale. This is S is equal to 10, green one, and 15, that is the scale value 15 is the red one. So this is the scaling of your dB7 mother wave. So here, this is just an example. You can do the analysis, single frequency CWT analysis. This is what you can do it in the octave. You can go for the range of frequencies. You can go for the logarithmic scale. But what I have done, and I got the good result, is with the single frequency CWT analysis, because it was possible in my case. It was possible in my case to identify the frequencies which are of the higher interest. So here you can see the low. So you can choose, you can specifically go for these frequencies and you can do the analysis. So here CWT analysis at the low frequencies, you can see the very highest CWT coefficients are there. You can find here CWT analysis. This is at the 1 frequencies, 1 Hertz, 3 Hertz frequency, 5 Hertz, 6 Hertz, 10 Hertz. You can do this kind of analysis also by using the CWT. As well as in the high frequency region also. This is a high frequency region, that is a peaks. Here this is a peak. You can identify this frequency, single frequency, and at this single frequency also you can do the CWT analysis. You can design your mother wavelet and you can do the analysis. So this is also CWT analysis in the high frequency region. These are the graphs. You can get multiple features from these graphs, and you can, and whether that features is contributing to your output or not, that you can identify. So this is see the true positive, false positive rates, and this is see this results that I'm showing to you, because single frequency CWT analysis also contributes a lot. Instead of going for the analysis of the all frequencies, you can take the major frequencies and you can do the analysis. You can find the true positive rate, false positive rate, precision, and depending on that you can choose the frequency that you want to only go for the final analysis of your EMG signals for the classifications or for the feature selection. So here the CWT at the 7 Hertz of frequency is contributing. CWT at the 8 Hertz of frequency is contributing a lot. So you can only choose this now. Instead of analyzing the whole CWT, only you can analyze this single waveform which is at the 7 Hertz or 8 Hertz of the frequency. This is for the CWT. Now coming towards the discrete wavelet transform. In discrete wavelet transform, what happens? Your whole signal, your total EMG signals will be divided into 2 frequencies. You have to downsample it. That is frequency, this, see this. Frequency is ranging from 0 to pi radians. So here it is divided into high frequency region and low frequency region. So here it is pi by 2 to pi, and it is 0 to pi by 2 radians. So here it is now again downsampled once again, and this 0 to pi by 2 again is divided into high frequency components and low frequency components. So this is your level 1. This is a DWT coefficients that we are getting over here. So pi by 4, pi by 2, 0 to pi by 4. Again you can downsample it and divide into, go to another level, that is a level 2. At this level 2, pi by 8, pi by 4. At this level 0 to pi by 8. Likewise, you can go to the down level, unless and until you will not get the features that you want for your classification or for your application. Unless and until you have to go towards the downsampling. So here what happens in this downsampling, your number of samples get reduced by 2. Yeah, suppose 200 the, so now it is 100 samples here, from here 150 samples, 50 to 25 samples. So your number of samples get reduced, and this may contribute in the degradation of your signal actually. But here, so this is your discrete wavelet transform. What happens in the discrete wavelet transform? Like in CWT analysis, you have to analyze the each and every scale. So here, your number of calculations are very, very lesser over here in the discrete wavelet transform. So see, this is a example from this paper of the discrete wavelet transform. You can see this EMG signal is there at the level 1, which is 0 to 75 hertz. This is the EMG signal, which is downsample. We divided into two parts, which is 0 to 375, 375 to 750 hertz. This 375 is also divided into two different frequency ranges here. So either here one high pass filter and low pass filters are used at each and every stage. And here you see they have gone for the level 7. So this is where you have to go, at which level you have to stop, and at what level you are going to get the signal. This is just a trial and error. You may get some things, that is a mother wavelet that you want to use, at which level you will go for from the literature survey, but still for your application, for your results, you have to do this analysis by your own. You can see these diagrams over here. See this EMG signal once again. Here this is D1, D2, D3, D4, D5, D6 and D7, and this is the highest. This is 0 to 6 hertz and this is 6 to 12 hertz. You can see this, the D1 which is there is 375 to 75 filtering of your signal. This is D2, D3, D4. As soon as we are going towards the D7, our frequency it reduces. So D7 it is 0 to 6 to 12 hertz of the frequency is there, and A7 is 0 to 6 hertz of the signal. If you'll combine, if you want to again get your EMG signals, so then at the time this X is equal to total this A7 plus D7 to your D1. D7 to D1 is your total EMG signal. So here discrete wavelet transform is majorly, most of the people they used for the filtering of the signal. So here I just want to show you some representations of the EMG signals, how you can represent your features. This is very basic feature representation. I have shown it for the myopathy, neuropathy and healthy signals. So here the 7 signals are there, these are healthy. This is your myopathic condition, and this is your neuropathic condition. So this is a representation of signal for the healthy conditions, for neuropathic condition and for the myopathic condition. This is another representation. This is another representation of your signal, which is a scattered plot of the prominent features. So what you can do, if you have captured some features, then first thing that you can do is you can go for the statistical analysis of the features. That is, you can find out its correlation value. If it is directly correlating with your output, then it is very good. But you can go for the t value, you can go for the ANOVA test, you can go for the p value, and you can find the prominent features. But still, if you are not getting anything, then you can go for the scattered plot for the prominent features. So here, this is what the mean value. So this is for one muscle activation condition. This is for the another muscle activation condition. This is the overlapping rate. You can go for the scattered plots also. This is another example of the scattered plot. This is again another example. Here you are having the different types of scattered plot, and this is a see the overlapping region over here for the new feature value, for variance, for number of peaks, and for the mean value for one muscle condition and for the second type of muscle condition. Now the next type of plot in a Veka toolbox. Veka is a toolbox where you can select the features, as well as you can get the ideas about the classification algorithms, as well as you can get the ideas about your feature selection algorithms. Plotting of your features with each and every feature also can be done in this Veka toolbox. So very good tool actually. So here also you can plot your features. So here the peak to peak amplitude is plotted for the healthy muscle condition and for the COPD muscle conditions. This is also a feature plot. So by observing this plot also, you can select your feature, which is the most contributing feature and which is less contributing features. Can also be selected by using the Weka toolbox. Now coming to the box plot. Box plot is also one important plot of your features, which can give the idea that which features you can select and which feature you can eliminate. Or you can have the feature selection algorithms. You can design your own feature selection algorithm. You can take the ideas of the feature selection algorithm, variance analysis, its correlation analysis can be done to select the variance features. Principal component analysis also can be done to select the features, or various feature selection algorithms can be combined together in the layers to get the feature selection algorithm. So, but before going to doing that, this box plot of the features will also give visual ideas about that whether that features can be selected or not selected. This is a box plot for the first grade, second grade, third grade, and fourth grade. Okay. The features, time domain, frequency domain, and time frequency domain features. It's a feature selection algorithm, classification algorithm. I'm not discussing now. I have feature selection algorithms. I have shared my email ID as well as phone number. In the first slide, you can contact me. The database, major problem of the biomedical signals, that is specifically for the EMG signals, is availability of the database. So here I have given some websites. This physionet.org, physiobag. So from here you can get the data of myopathy, neuropathy, and this healthy muscle conditions. Here also for different, like I said that for different electrodes, how your EMG will look like can be get through this EMG.org. RamiKushaba.com. This Kushaba sir has also given his own database for the students, as well as this is a data bank, as well as you can contact to the hospitals. They are